Hey guys, what's up? My name is Luke Osborne. I'm an undergraduate here with Kurouski Laboratories. One of the roles that I play in this lab is protein purification of these amyloidogenic proteins that we work with. Now we've developed several methods in this laboratory for purifying these proteins. We have ion exchange chromatography, affinity chromatography, size exclusion chromatography. But one of the most promising methods that we found works for these amyloidogenic proteins to purify them is called ammonium sulfate precipitation. So once we do this procedure and we've got all of our proteins purified, we can pass it along and carry on with many other kinds of experiments with these protein products.
Howdy, this is Abid Ali. I'm working as a postdoctoral fellow in Kurouski Labs. My research work mainly focuses on to investigate the influence of the lipids and fatty acid, the proteins aggregate rate, secondary structure changes, and toxicity of the amyloid, oligomers, and fibrils. To fulfill my objective of my project, we develop many approaches to like techniques, cloning, and site-direct mutagenesis to make the many mutations in particular protein alpha-synucleins and transthyretin to replace the many specific amino acid to another amino acid and check the particular effect of the these particular amino acid with the lipid and fatty acids.
Howdy, I'm Michael, a PhD student conducting research in the Kurouski Lab, investigating impact of lipid vesicles and fatty acids on the rate of protein aggregation is a massive part of our scientific pursuits. To facilitate these investigations, we employ state-of-the-art plate readers. These instruments enable precise control of incubation, agitation, and continuous monitoring of fluorescent probes used to detect amyloid fibrils at minute intervals. This cutting-edge approach allows us to discern subtle changes occurring at each stage of aggregation, offering valuable insights into how different fatty acids and lipid vesicles influence the aggregation kinetics. Our findings pave the way for understanding the intricate mechanisms underlying protein aggregation and its modulation of various lipid species. In this cutting-edge cell room, our research focuses on how altered amyloid protein aggregation impacts mammalian neuron toxicity. We conduct tests on cell death, mitochondrial stress, and reactive oxygen species to see how these protein aggregates impact when aggregating in presence of different lipids. These assays provide valuable insights into cellular health, allowing us to track toxicity on an organelle level and understand how protein aggregates affect cell well-being. Join us as we explore the mysteries of neurobiology and work toward finding solutions for neurodegenerative diseases.
Hi, I'm Tianyi. I'm a fourth-year PhD candidate in Kurouski's lab. My primary project is understanding the role of lipid in alpha-synuclein aggregation process. So amyloid protein aggregation is a very heterogeneous and complicated process. Utilizing nano-IR, we can do single particle analysis, so we can know the structure of oligomers, fibrils, and we will link the structure with their cellular damage. Our research can provide an insight of which lipid biosynthesis pathway to target in resolving the neurodegenerative disease. With their help, we can solve this problem.
Hey, I'm Axel Rodriguez. My focus is on studying the proteins we express through nano-IR using atomic force microscopy. With this instrument, we can observe the protein morphology, mainly their secondary structure, to understand their development in amyloid protein aggregation. Here we have the instrument, the nano-IR3 from Bruker Nano, which is a combination of AFM and IR spectroscopy. It enables single particle characterization, where we can distinguish the different protein aggregate conformations. Utilizing this technique, our aim is to answer the role of lipids in the protein aggregation process and to relate the protein secondary structure with their cellular toxicity.
My name is Dmitry Kurouski. I'm associate professor in the Department of Biochemistry and Biophysics. In my group, we have 4 independent and interconnected research projects that are focused on the plasmonic catalysis, forensics, detection and identification of biotic and abiotic stresses in plants, as well as elucidation of the underlying molecular causes of neurodegeneration. I would describe myself as the hands-off mentor that does not like micromanaging. At the same time, I meet with my students bi-weekly to track their research progress and discuss science. And I think these bi-weekly meetings are very important because they help to avoid procrastination, and at the same time they give an opportunity to students and my postdocs to understand how well they do in the lab. In addition to bi-weekly meetings, I ask my students and postdocs to present their research on our weekly meetings, and considering the number of people in my lab, a student or a postdoc typically presents once in about 2 months.
Here in the Kurouski Lab, our forensics team is formed by students from diverse studies such as toxicology, entomology, environmental sciences, and, of course, forensic and investigative sciences. Our research spans even further in areas such as forensic entomology, forensic hair analysis, controlled substances within plants, and much more. For each of them, spectroscopic methods such as Raman spectroscopy and infrared spectroscopy are used to reveal, identify, and differentiate each specimen. Our research doesn't even stop after the paper is written. We also present at several conferences which help us build skills such as communication, networking, and expertise.
Spectroscopic techniques take advantage of the interaction between molecular bond vibrations in visible and infrared radiation. Infrared spectroscopy measures the number of photons that are absorbed by electrons within molecular bonds by irradiating a sample with the wavelength in the infrared spectrum. Raman spectroscopy measures the number of photons from the visible spectrum that are inelastically scattered by electrons within molecular bonds. There are two types of scattered photons that a Raman spectrophotometer can collect. One that reflects with a higher wavelength, which we call Stokes scattering, and photons that reflect at a lower wavelength, called anti-Stokes scattering. It's more common to measure Stokes scattered photons. However, since they occur at a more frequent rate than anti-Stokes, the Raman overall is a relatively weak phenomenon.
Most of our forensics research is conducted using this giant thing. We utilize a home-built Raman confocal microscope to adjust the surface area of our scans, so each spectrum is high in intensity and possesses very little noise. Using light field, we can also adjust our acquisition time, which allows us to control the number of photons that reach the sample during each scan. Today, we're scanning dyed hair using surface-enhanced Raman spectroscopy, a technique that utilizes plasmonic solutions like gold nanoparticles to increase the number of scatter events. Our Raman spectra have two variables: the intensity, which is directly proportional to the number of photons collected, and the Raman shift, which is a derivative of the different wavelengths photons are scattered at. In this scenario, we can tell that wavelengths are longer than the original since our Raman shift is positive, indicative of Stokes scattering. The other benefits of using confocal are that we can change the Raman shift to wherever we like, as well as adjust the power of the laser that reaches our specimen using filters.
Also using Raman spectroscopy, fibers are another material that can be used using a handheld Raman spectrophotometer. The handheld is helpful when using it in the field because it's lightweight and portable to different places. This is useful to having to move between different crime scenes and labs.
One of the experiments we run using infrared spectroscopy is analysis of forensically important fly larvae or maggots. Using a Fourier-transformed near-infrared or FT-NIR spectrometer, we're working on identifying the differences in chemical compositions of fly larvae, such as differences in protein, lipids, sugars, and several other compounds. One of the applications for this research includes offering a non-destructive method for analyzing insect evidence so it can be used again later.
Hey, my name is Isaac Juarez. I'm a second-year graduate student in the Kurouski Lab. Part of my work focuses on synthesizing various nanoparticles for application in various projects around our group.
Hello, my name is Swati Patel and I'm currently working as a postdoctoral researcher in the Kurouski Lab. My research mainly focuses on the development of nanomaterials for the plasmonic-driven photocatalytic application. Nanomaterials research approaches nanotechnology from a material science perspective, leveraging advances in materials, metrology, and synthesis that emerged in microfabrication research. Our lab prepares plasmonic nanomaterials to different chemical and physical methods. Considering nanomaterials with nanoscale structures often have unique optical, electronic, and thermophysical and mechanical properties. So we have prepared gold, copper, palladium, and other nanomaterials with different shape and different size. And in addition, in our lab, we are focusing on to prepare the different nanomaterials like carbon, nanotubes, graphene, or some metal dichalcogenides like molybdenum disulfides, tungsten disulfides.
Plasmonics is a technical branch of photonics that focuses on the interaction between light and nanomaterials. At the core of plasmonics lies the fundamental physical principle of localized surface plasmon resonances, also known as LSPRs. These are coherent oscillations of conductive electrons on nanoparticle surfaces. In plasmonics, nanomaterials are integrated with light source, such as a laser, to study and manipulate molecules, leading to enhanced light-matter interactions with broad applications in various fields. One way we apply this in our lab is through surface-enhanced Raman spectroscopy. abbreviated as SIRS. SIRS amplifies the Raman signal of an analyte by several orders of magnitude, allowing us to look at analytes in extremely low concentrations. Our forensic research uses nanoparticles to look at artificial colorants present on hair strands, while our plant research is aiming to study the benefits of SIRS in diagnosing pathogens and stressors in plants. We primarily use our confocal microscope in SIRS. However, other groups in our lab use TIRS, combining scanning probe microscopy and Raman spectroscopy technology.
TIRS, it is evolutionary advancement in vibrational spectroscopy of nanometers, with spatial resolution and sensitivity on the order of 10 nanometer. TIRS dramatically improves the Raman signal by employing a sharp metal tip similar to that used in airfare, resulting in a highly immersed field at the apex. The apex of the tip generates a highly amplified field, which when positioned above a molecule enhances the Raman signal at the nanoscale, and simultaneously we can record the surface topography of the nanomaterial. TIRS introduces a new dimension to SIRS by enabling two-dimensional chemical imaging with nanometer optical resolution. Plasma-driven catalytic reaction as a part of photocatalysis has attracted immense attention. So due to the collective acceleration of free electrons at the surface of metallic nanostructure, the charge distribution stores energy from the incident light that could transfer energy to the molecule that promote the photocatalysis. So our study uses the plasma-driven photocatalysis of benzone-containing group like nitro, bromo, and chlorothiophene on a different plasmonic nanostructures under the ambient conditions. So as an environmental friendly and green photocatalysis process, plasma-driven reaction illustrates a brilliant future.
Our work in plasmonics has garnered recognition on both local and international stages. We have presented our findings at prominent events, such as the International Conference on Surface Plasmon Photonics, hosted by Rice University this year. The field of plasmonics has experienced an exponential surge of innovation since 2010, opening the doors for exploring novel applications of nanoparticles, SIRS, and TIRS. Given the rapid growth in this domain, the expertise I have gained in this field holds significant value and is highly sought after in the industrial sector. Seeing that plasmonics can be applied to almost any field, it is a skill I would recommend anyone coming through our lab to obtain. The primary work I do revolves around using Raman spectroscopy to diagnose various biotic and abiotic stresses within crops. Several undergrads helped me with these projects, and I've also had the opportunity to assist with their own projects. In addition, I utilize high-performance liquid chromatography, also known as HPLC, and inductively coupled plasma mass spectrometry, also known as ICP-MS, for further investigations.
Besides forensics, I apply myself to plant science as well. In plants, I use Raman spectroscopy on hemp and HPLC analysis on tomatoes. Involve myself in different fields of studies, it's important to connect these fields together.
With the growth of the human population, food demand will increase by 30% in under 30 years. Furthermore, anywhere between 30 to 40% of crops are lost annually due to pests and environmental stressors. Therefore, protection of our crops is of vital importance to maintaining both the global economy and ensuring there's enough food for everyone. We propose to tackle this issue with Raman spectroscopy. In our lab, we have evidence that Raman can be used to detect viral, fungal, and bacterial diseases in a large number of plants, along with abiotic stresses such as salinity, arsenic, and other heavy metals.
This technology has been successfully utilized in previous projects. Among them, we've been able to detect the hormone-bean virus in citrus crops, identify the tomato spotted wilt virus in tomato crops, and effectively assess salinity stress in stalkroot in maize crops.
Presently, my research revolves around exploring Raman spectroscopy's potential to diagnose arsenic stress in rice crops. Our findings have revealed that Raman spectroscopy can detect toxic levels of arsenic in rice before visible symptoms begin to manifest in the crop. This investigation holds a special significance considering rice's vital role as the staple crop for over half of the world's population.
Using the handheld Raman spectrophotometer, we've been able to detect and characterize different types of stress that hemp experiences before visible signs of stress appear. This makes growing hemp easier and cost-effective when specific stressors such as light, heat, or water stress are experienced by the plants.
Overall, this technology is easy for both students and private sector employees to use, and there's lots of room for improvement and innovation. 5 years ago, we couldn't have estimated the amount of work that our group would have to invest into developing this technique, and working at the forefront of a field is one of the most exciting opportunities I've had in the Kurouski Lab.